In today's class, we are going to discuss another post-fertilization event, and that is how zygote would develop into embryo. Alright. Now, if you remember in the last class, we have discussed how it is very much important that first it should be the pen developing into endosperm. Right? Because the endosperm is the food-laden tissue that should be ready to provide nourishment to the developing zygote. Right? So in that case, always pen first will develop into endosperm. Then it is zygote that would develop into embryo. Right? Now zygote developing into embryo will be discussing under two sections. In the first section, we'll discuss how dicot embryo develops, and its second section will be discussing how monocot embryo develops. So let us start with dicot embryo development. Alright. Now, first of all, for dicot embryo development, you have to recall the structure of ovule. The most common type of ovule being anatropous ovule or inverted ovule. This being the micropyle, this being the salazar, isn't it? And this is the embryo sac. Alright. So after fertilization, towards the micropyle remains the zygote, and here the entire space of the embryo sac is going to be filled up with nutritive tissue. The tissue which we call as endosperm, and which is now ready, right? Which is ready and fully developed, and now the zygote will start to show its development, right? So let me mark the zygote here. So this is the zygote, which you all very well know. It's a diploid structure. And throughout our discussion, one thing is going to be very important for you to understand, that we will mark here the direction of the micropyle and salazar. Because if you don't remember the orientation of micropyle and salazar, in that case the entire idea of how the zygote will develop, right, that can become completely wrong. So in that case the orientation is very important. So let me mark here. Suppose this is the micropylar end and this is the salazal end. Alright. Now, another important thing again that you have to remember throughout our discussion is that the zygote, when develops into embryo, it's going to continuously show cell division, right? And the cell division here will be occurring by mitosis. So, the entire embryo or all the cells present in the embryo will be diploid. Fine. So remember here, the nutritive tissue, that is the endosperm which has developed, is triploid, whereas the zygote finally will develop into embryo. The embryo will possess all cells diploid. Okay. So it's by mitosis that the entire zygote will fully develop into embryo. Alright. So let's see how the development will begin. Now, first see, the zygote will undergo one round of transverse division. Okay. So, one round of transverse division and unequal division. One round of transverse division, which is also going to be unequal, because it will give rise to two cells. both the cells will have two different fates. That's why I am using two different colors to denote them, so that it becomes easy for you to understand that which particular cell is giving rise to which part of the embryo. Okay. So this being the micropylar end, this being the salazar end. See, towards micropylar end, the larger cell will be formed. We will call it, will be termed as basal cell, whereas the cell towards salazar will be known as embryonal cell or terminal cell. Alright. Now, first let me show you how the basal cell will show its division. The basal cell will undergo a lot of transverse division. Okay. So, if it undergoes a lot of transverse division, many cells will be formed. Okay. Many cells are formed. How are they formed? This one cell will undergo one transverse division, giving rise to two cells. Then this two will again divide by mitosis, giving rise to 4 cells. Likewise, they will divide. Okay. And form a long stalk-like structure. This long stalk-like structure is going to be known as suspensor. Okay. And the last cell or the cell of the suspensor that remains towards the micropyle, that will become enlarged, and we will call it the hostorial cell. Hostorial cell. Alright. The terminal or embryonal cell, it would remain as it is. Okay. Now, the suspensor or the hostorial cell, it will remain as it is. It will not divide further. Further, the changes will start occurring in the embryonal cell, that is towards salazar. So, let's see what are the changes that will take place in the terminal cell. The suspensor will remain as it is now. Okay. So this being the micropylar end. Now, first of all, the terminal cell will undergo one round of longitudinal division. As it undergoes one round of longitudinal division, two cells will be formed, and these two will be termed as proembryo. Proembryo. So this is two-celled proembryo. Alright. Now, next. Next, what would happen? These two cells which have been formed by longitudinal division will again undergo one round of transverse division. So, if it undergoes transverse division once, now how many cells will be formed here? 4 cells will be formed. So, we will call it proembryonal quadrant. 4, that's why quadrant. Okay. So, this is the quadrant stage of embryo development. Next. Next, what would happen? Again, as usual, suspensor will remain. Alright. These 4 cells will undergo another round of division. So 4 cells undergoing mitosis will give you 8 cells. So this will become further enlarged and will give rise to 8 cells. Alright, 8 cells will be formed. Now, this will be known as pro-embryonal octant. 8, so octant. So this is the octant stage. Alright, so as usual, micropyle salazar, micropyle salazar. This is micropyle salazar. Okay. Now, one thing here all of you should remember. See, these 8 cells that I have drawn here, it's actually not going to look like this. Okay. It's a schematic diagram. That's why it is like, for making it easy for you to understand, I am making the 8 cells like this. But actually, the embryonal cell, when it starts to divide, it divides in all the planes. Okay. So suppose if there are 8 cells here I am showing, so few cells will be towards front, few cells will be towards back side. So it will be like a mass of cells, like a ball of cells that will be formed. But a ball of cells I of course cannot be showing you here, right? So that's why, to make you understand properly, I am drawing all the 8 cells like this. But otherwise, while it actually happens, it will be happening in all the planes. So the cells will be in the form of a mass, in the form of a ball-like structure, globular structure. It won't be like this. Alright. Now, what would happen, you see, these are the 8 cells of the embryo, right? Now, they will show periclinal division. So periclinal division means it will be giving rise to one cell towards outside and one cell towards inside. Okay, while the cell division happens, one cell will be deposited towards outside and one cell will be towards inside. So how will it look? Suppose this were the 8 cells, right? These were the 8 cells, right? Now while the 8 cells will undergo periclinal division, so one cell will be towards outside and one cell towards inside. So like this, a layer of 8 cells will be formed towards outside. So we have how many cells in total? 16 cells, right? 8 cells are towards outside and 8 cells are towards inside. That much is clear, I guess. So this outer 8 cells will form a layer called dermatogen. Dermatogen. And dermatogen will later on give rise to epidermis. That is the outer skin of the plant body. So as usual, this is the micropyle and this is the salazar end. Okay, orientation is very important for you to remember. Now, the next change that will happen. See, the next change that will happen is in one cell of the suspensor. See, this was the entire suspensor, right? So one of the cell, this one of the cell, this cell, that is extremely towards or that is in contact with the proembryo, that will now start to change. Okay, and we will call the cell that it will be changing into to be hypophysis. So it will look like this. This is the proembryo, right? Right? Now, this cell, the cell that is closest to the proembryo, the cell of the suspensor which is closer to the proembryo, that will change, and we will call it hypophysis. Hypophysis, okay? Rest of the cells will remain as it is. This is micropylar end, this is salazar end. Now, next change that will happen is in the hypophysis, because now the hypophysis will start to divide continuously, and the hypophysis will also form a mass of cells. Few cells towards towards the front, few cells towards the back side. But because again that will make it difficult for you to understand, I am going to make all the cells together. So it's going to look like this. The hypophysis, it will divide and it will give rise to 8 cells. Okay, 8 cells will be formed by the division in the hypophysis. Suspensor will remain as it is. This is micropylar end, this is salazar end. Okay, now before I go further and show you what kind of changes will further occur in the embryo, you must know that which of the cells will give rise to which part of the embryo exactly. So what is the fate of these cells? That is very important for you to understand or remember. Okay, this is here. Alright, let's see. See, the 4 cells, these 4 cells of the hypophysis that are towards the micropylar end or that are towards the suspensor will give rise to root tip and root cap. Very important. Alright, for you to understand this is very important. Whereas the 4 cells of the hypophysis which are towards the proembryo or towards salazar, that will give rise to root cortex. Okay. Now, the lower half of the proembryo towards the micropyle will give rise to hypocotyle, whereas the upper half will give rise to plumule and cotyledon. Plumule and cotyledon. Alright. So these are the different parts of the embryo that will be formed from this particular cells. Knowing this, now let us see what are the changes that will finally be happening in the embryo. Now, as you know that the root tip, that means root tip, root cap, root cortex, all of the parts related to root is actually then being born from hypophysis. Do remember. That means in the embryo, the root will be formed or the radical will be formed from the hypophysis. Okay, that's the first thing. So when these cells will continuously divide, they will form a mass or a tissue, isn't it? And that is going to look like this. it will give rise to an extension, a radical-like structure. So suspensor is going to remain as it is. Alright. Now, you might be thinking about this proembryo. So, proembryo will also start to continuously divide and cell division will occur, and that's when it will do a structure like this. You might be thinking it's a heart-shaped structure, isn't it? Exactly, that's what is going to happen. So see, the hypophysis, it has continuously divided and has given rise to a mass of cells which has been extended and formed the radical-like structure or the radical. So this would be the tip of the root. So this is the radical, isn't it? Whereas this proembryo, it has continuously divided and has given rise to another mass, like this part, the lower half of the proembryo will be here as a mass of cells, giving rise to the hypocotyle, whereas the upper half will also divide and they will show like little more of division. Cell division will be more in these two regions. So these two will form the two cotyledons. It is dicot embryo development that we are discussing. So that's why it will form these two extensions are the two cotyledons. And from here exactly comes out the plumule. This is the plumule. Alright. So see, at first the dicot embryo will be circular. When it is continuously dividing, it will give rise to a circular or a ball-like embryo. This ball-like embryo we call as globular embryo. Globular embryo. Later on, by continuous cell division, when it takes up a heart shape, we call it heart-shaped embryo. Alright. But the cell division or development does not stop here itself. These two extensions, they will continuously further, due to cell division, they will keep on growing like this. So while they are growing, they will not remain straight like this. They will instead fall off. So like this. As if it is growing and it is going to fall off like this. And this is also growing, this will also fall off like this. Okay. So finally, as it falls off, how is it going to look like? See, it is going to look like this. Like this. The cotyledons will extend. The cotyledons will extend and will form a structure like this. Here will remain the plumule. This is the radical. This is the radical. This is the plumule. And these are the cotyledons. So finally, when fully developed embryo will be formed, it is going to look like this and we call it horseshoe-shaped. Horseshoe-shaped embryo. Alright. So this is the entire part or entire thing how a dicot embryo will develop. Alright, these are the various steps of dicot embryo development. Okay, so let me revise it again for you so that it becomes more clear. So first of all, this is the diploid zygote which is towards the micropylar end of the embryo sac. Okay, the zygote for developing into embryo will undergo continuous mitotic division. So in the embryo all the cells are diploid. Okay, so see, first of all the zygote will undergo one round of transverse division which is going to give rise to two unequal cells. So one of the cell towards micropyle, that is larger, will be known as basal cell, whereas the one which is towards the salazar, that is smaller and called as terminal or embryonal cell. Okay. Now, the basal cell is going to give rise to the suspensor, whereas the embryonal cell or terminal cell will give rise to the embryo. So both the cells have two different fates. Okay. So first of all, the basal cell will undergo continuous transverse division and will give rise to a stalk-like structure, and this stalk-like structure is called suspensor. The cell of the suspensor, which is towards extreme micropylar end, will become enlarged and will be known as hostorial cell. Okay. So by this, the suspensor has fully developed. It is not going to change further. Okay. Now, let's see what will happen in the embryonal cell. In the embryonal cell, first division that takes place is a longitudinal division giving rise to two cells. So these two cells will be known as proembryo, and this is the two-celled proembryo that has been formed. Now, the two-celled proembryo will undergo another round of transverse division and give rise to four cells. So four cells, we call it proembryonal quadrant or quadrant stage of embryonal development. Okay. Further, this proembryo will start dividing in all the planes, giving rise to a ball-like or a mast-like structure. Okay. So few cells will be in the front, few cells will be at the back, likewise a structure will be formed. But for better understanding, I am making the diagram like this here. Okay. So now four cells will undergo another round of mitotic division giving rise to 8 cells. So we call it the octant stage. Okay. Or the proembryonal octant. Okay. Now, these 8 cells will undergo now one round of periclinal division, means one cell will be deposited towards outside and one cell towards inside. So if that happens, 8 cells will be formed towards outside and 8 cells as usual are inside. The 8 cells which are formed towards outside, that is going to be called as dermatogen. And dermatogen is the part from where epidermis of the embryo arises. Means from here, the outer skin of the embryo will be formed. You know very well what is epidermis. So the outer skin of the embryo will be formed. Okay. Now, so see now, after the dermatogen has developed, finally one of the cells of the suspensor will start to undergo a change. And which cell will undergo the change? The cell of the suspensor which is attached or which is the nearest to the proembryo. That will start changing into hypophysis. Hypophysis is that cell from which the radicle or root of the embryo will develop. Alright. So see now, the hypophysis will also start undergoing continuous division giving rise to a mass of cells. But then these cells will have different fates which I have showed in this diagram. See. The 4 cells of the hypophysis which are towards micropylar end, that is going to give rise to root tip and root cap, whereas the 4 cells which are towards salazar, that is going to give rise to root cortex. So in short, the hypophysis will form the entire radicle of the embryo. On the other hand, the cells of the proembryo towards micropyle, that will give rise to the hypocotyle, whereas the upper half of the proembryo will give rise to plumule and cotyledon. Alright. So fate of each of the cell is known. Now which part of the cells will give rise or which cells will give rise to which part of the embryo, that is well known, right? Now see what will happen. Finally, the hypophysis will continuously divide and form a mass of cells which will extend and form a radicle or a root tip. Alright, so it is going to look like this. On the other hand, this proembryonal cells, they will also continuously divide and form a mass or a tissue of cells, and that is going to show two extensions. Okay, on two sides of the proembryo, there will be a little bit more of cell division, giving rise to two extended parts. These two extensions, they will be known as the cotyledons. And from in between the cotyledons arises out the plumule. Okay, and this part is the hypophysis. Alright, so in the first place, the embryo is round or circular. We call it the globular embryo. But later on, when the radicle has extended and cotyledons and plumules have started to arise, it will look like a heart shape. So we call it heart-shaped embryo. Finally, the last stage is when the cotyledons will grow and they will start bending like this. So when they bend like this, the dicot embryo will finally look like horseshoe-shaped. Alright, so in that case, the embryo is first globular, then heart-shaped, and then horseshoe-shaped. And this is how it looks at the final stage of development. Okay, so I hope all of you are clear about it. Now for a very important question. This entire thing being discussed, what was the main purpose of this suspensor? What is the need for the suspensor? Look at this diagram here. We have drawn here an anatropous ovule, inverted ovule, isn't it? See, now this being the nutritive tissue, endosperm, isn't it? So this is the suspensor, right? And this being, suppose, the embryo. Okay, the red colored part is the embryo. So the suspensor is actually holding the embryo and it is pushing the embryo towards the nutritive tissue. That means towards the endosperm. So it is just like the suspensor is that part with which the embryo will remain suspended, and its function is nothing but to push the embryo deep inside the endosperm so that the embryo can take more and more nourishment from the endosperm and then it can develop. Alright, so this being this function of suspensor, and this entire thing being the development of dicot embryo, we complete the class here itself. In the next class, we'll discuss the monocot embryo development. Thank you.